Good evening, everyone. Welcome to Genotypic's webinar series. Thank you for joining us today. I am Saroja, General Manager, Business Unit at Genotypic Technology. I will be moderating today's session on why choose long reads over short reads for transcriptome sequencing. I have Dr. Meenal, my colleague, a principal scientist, heading a Nanopore lab and analysis team, joining us to answer your queries on chat. Hence, please feel free to drop your questions in the chat box at any time during the session. Before we begin the session, I also request all the participants to kindly keep your mics muted throughout the presentation and turn off the videos. Now, it's my pleasure to introduce today's speaker, Dr. Raja Mugasimangalam, CEO, Genotype Technology. Dr. Raja is a veteran technocrat and entrepreneur in the genomics sector since 1998. In addition to Genotypic Technology, which is India's first genomics company, he is also the founder of Qutilomics, India's first agri-genomics company. Applying genomics in real-life problems in research, product development, and testing are his expertise. Dr. Raja is an elected executive committee member of ABLE, Association of Biotech-led Enterprises of India, and takes care of networking, public relation, and works closely with startups and startup incubators. He is also a member of Board of Studies in many universities for genomics, genome informatics, and bioinformatics, and also of several professional organizations. He is also an active publisher and reviewer in peer-reviewed journals. He runs the world's largest genomics LinkedIn group with more than 1 lakh followers. Now, I warmly welcome Dr. Raja to take us through why we should choose long reads over short reads for transcriptome sequencing. Thank you.
Thank you, Saroja, and thank you, everybody, for this webinar. So firstly, let's see who are here. Around 660 registrations came, most of them from India, but I'm glad around 15% are coming from different parts of the world. Welcome, everybody, at different time zones. And people who are not able to join now at this time, we are going to send the video link, recorded video link. And for your information, AI is listening, artificial intelligence, and hopefully AI might summarize better than what I'm telling. So directly coming to the point, these are all the input which you all gave. Thank you very much for it. Most of you are doing, as you can see here, using short reads, but good number, almost 15-20% are using NGS long reads, microarrays, 15% still using, RT-PCR. I think RT-PCR is the final confirmatory test. And thankfully, almost half of you are not yet started. So I think this is because almost that 350% are students. So I'm glad I'm starting you early into a better technology than using short reads. But there are professors, there are lab heads, technicians, scientists, clinicians. I welcome everybody and thank you for joining this webinar. So brief history of the journey of Genotypic. We started in 1999 when the company was incorporated. And then we brought genomics to India. At that time, we were the only or maybe the second company which was offering genomic services. Otherwise, genomic services were offered by only core facilities in research institutions. Then we became Agilent Certified Service Provider, Iron Torrent, SAP, Illumina CS Pro. Then we got into Nanopore. UK, Oxford Nanopore Technologies, and we fell in love with them immediately because the reads were long. Even one read tells something. When you do a microbiome, just one read tells something because the read is long enough. Then this year, we also came into ONSO, PacBio's ONSO, which is the most accurate Q40 sequencer, which we already have in Genotypic. We found that this Q30, Illumina, MGI, and other things, I think the deficiency of short reads was compensated by the Q30. Why Q30? We can now go for Q40, which is 1 in 100,000. Q40 to Q50 is accuracy or error rate of 1 in 100,000. This is really make a huge difference for many applications. Now back to about Genotypic, what else we are doing. We have a team of 70 plus, and we have two more companies, Qtelomics and Dithyomics. Qtelomics in healthcare and I'm sorry, agriculture, and Dithyomics in diagnostics, totally around 100 people. So we have more than 650 publications coming out of our work to people like you. So we do lots of offerings in genomic services. This is from year 1998. The new ones are here, Q40 to Q50 reads, Nanopore, high throughput. We have multiple machines. We still use Agilent microarrays. We have 10x Genomics, SomaLogic, and also platforms for single cell genomics. 10x is for single cell transcriptomics. So let's come to RNA sequencing. Why we do RNA sequencing? The first and foremost is RNA-seq came in and replaced or replacing microarrays. This is for gene expression. What genes are upregulated upon a disease and what genes are downregulated in a disease situation or when you give a drug. So this is up and down regulation is the major application of RNA sequencing. This also improves genome annotation. Like you thought there is a gene and now you're finding the transcript. And this is for non-model species, sometimes you only do RNA sequencing, not genome sequencing, so that you have the reference RNA-seq or reference RNAs sequenced. Gene fusion, splice variants is not only for cancer, for almost everywhere we require this. And this is where long reads are more important. This and this genome annotation, you can already see, except for gene expression, which also I'll come down and say, and then novel non-coding or long RNAs, even coding RNAs which were never known. For example, in chicken, some of the genes, important genes were never sequenced using short reads. After long reads, they all basically appeared. Of course, you can do molecular markers, SNPs, SSRs, QTLs. I have another place I'm going to describe that. This is why you do RNA sequencing. It has become a very important tool. So how do you plan an RNA-seq experiment? So you start with experimental design. What is your biological question, right? If the biological question is, I'm treating a cell line with a drug, right? So then the type of sample is cell culture. Now talk about replicates. Untreated, treated also, there could be different dose you treat and different time points you treat. And there has to be biological replicates. Typically 3 replicates. So for example, if you have 2 time points, that's a 0 time point, and say after 24 hours, just 2 time points and 3 different doses. So that will become 2 into 3 into 3 of the replicates. The most important stuff, of course, is how do you extract the RNA and then the library preparation protocol. The problem with library preparation protocol with short read is you shear the DNA, RNA. RNA is sheared. right? And then there are PCR, multiple PCR steps before you make the library, after you make the library, and after you put it on the flow cell, another PCR, and then sequencing itself is like a polymerase mediated thing. And then you analyze. You QC the data, map the reads onto the reference RNA, and then you count. You count. And then you say how much is the gene expression. So this is where short reads versus long reads, which platform you choose is very important. With the short reads, the major problem is sometimes the same RNA, one part is upregulated, one part is downregulated. Obviously, there is maybe a splice variant, but you don't know because you only have pieces of RNA which has been sequenced. You don't even know how the full length is there. So this is the question, short read or long read. What will you use when? So there are two strategies. First one is the poly-A-based library preparation, where you have magnetic beads with oligo-DT, and then you capture all the polyadenylated RNA. And then you go along with your library preparation part. Or you remove the most abundant RNA, the ribosomal RNAs. Then you sequence the rest. In this case, non-polyadenylated RNAs can also be sequenced. So what is the end of a RNA-seq data? Firstly, one thing I'm not showing, which no one wants to see, is an Excel sheet, right? Excel sheet with reference RNA IDs and the name of the RNA and what is the expression values in each condition. So here are two conditions. So now we can see that these genes are upregulated, these genes are downregulated, and these are name of the genes. That's called heat map. Then you go and see the pathway, what these up and down regulated genes mean. Maybe glycolytic pathways improved, increased, certain catabolic pathways, adiponeogenesis or decreased. Like that, we can see what is going up and what is going down. And then you have correlation maps, bubble enrichment analysis, bubble plots. There are different ways of showing the same thing. But now let's come to the nanopore long read sequencing. Very quickly, there is no chemistry. It's interesting, right? There is no chemistry, like everywhere you say, this is a dideoxy sequencing with reversible di-terminator chemistry. There is no chemistry. Here is DNA or RNA, which is ligated to a motor enzyme, which is just a helicase. And then nanopore also is a porin, bacterial porin, modified, sitting on a membrane. And it is pushed at almost for RNA-seq 100 base pairs per second. And then electricity is measured because it passes through electric current. And then this RNA blocks the electric current. And depending on the bases and also base modification, the graph changes. So this is nothing but image of the or shadow of the DNA or RNA in an electric field. Interesting, right? There's no chemistry, it's physics or electricity. And it happens so fast. Even within 5 minutes, you would have sequenced a bacteria. Or in 5 minutes, you will start getting each barcode, how many reads have come and what are the upregulated genes. You can even see what is called as real-time analysis right away. So this is the most important slide. Why long reads? Why not short reads? You see here, exon 1, exon 2, exon 3, each exon is already split by all these tiny reads, right? And luckily it's padded, so we can all put together. And then you say, hey, here is one splice variant, there is another splice variant, and all it has to be mathematically calculated. Whereas in a long read, it is one single piece of cDNA. I can go and say, hey, boss, there are different isoforms. I have 3 different isoforms in that tissue. Whereas if you do these short reads, finding these 3 itself is a mathematical puzzle. And if any one isoform is expressed low, you may not find it at all. I think this slide clearly explains how different multiple isoforms gotten and then full length of the transcript sequenced. And there are 3 different ways of doing RNA sequencing using long reads. First is PCR cDNA sequencing. That is, in case you have very little RNA, not enough for sequencing, but you don't chop it down, even the full length after ligating to adapters, you do a small number of cycles, not heavy number of cycles, so that you don't disrupt. But it's better what is called as the middle portion, direct cDNA sequencing, which is PCR free. This needs almost 100 nanogram of poly A plus RNA, which means we have to start with a lot more, like 10, 20 micrograms of RNA. So if you have less of it, you go for PCR. But in most cases, what we have seen is we have enough RNA. And then direct RNA sequencing. Here, this removes even the bias created by RT-PCR, RT or PCR. So you can sequence RNA directly. I'll show you later how it works. But now coming back, see, one of the greatest advantage of short read sequencing is it is cheap. You can make a lot of pieces, right? I'll say I'll make 5 million reads, 3 million reads, like that. So you can see that this chart clearly shows that longer the fragment length, you do not need that many reads to understand the expression. Shorter it is, you need more and more data. So the complexity of a gene expression is usually lost in short reads. You'll only see which gene is more transcribed, which gene is less transcribed, and maybe which isoform, maybe the major isoform you will know. Whereas in a long read, you directly identify which isoform is present most. Now I'm going down to direct RNA sequencing. This is something amazing. We never thought we can sequence RNA directly without converting it to a cDNA and then PCR and then making library. So here you have to attach the ligate the motor protein to a single strand RNA, and then the RNA goes through the pore. So what's important here? We will know RNA modifications, which we would have otherwise never know. After you do a convert into cDNA, the modifications are lost. And then how much is the poly A tail? So you can say, or even the diversity of tails within the same transcript, same RNA can be seen. And it can do a complete isoform mapping, transcript quantitation, native RNA data, you will never get it. And it's faster because there is no batches, or you need not wait for cDNA library construction and things like that. See, nowadays, this unseen biological insights, a lot of genome institutes are running direct RNA sequencing for cancer samples, for tissue samples, so that they understand RNA methylation along with, of course, DNA methylation better. So what are the main things? This removes reverse bias created by reverse transcriptation, transcripts or PCR, and you know exactly how the RNA looks. And it's also important that some RNA do not, because RNAs have secondary structure. And this is why chicken, some of the RNAs never got sequenced, important RNAs of hormones, even lipid related hormones, adiponeogenic hormones were never sequenced until our direct RNA sequencing was done. And then, of course, the modified bases, now that's a new science going on. So if you do the nanopore, direct RNA availability right now is only that. It's the only difference is here is you will need much more RNA. Now the amount is getting decreased, but you require a lot more RNA than if you want to do a PCR-based cDNA sequencing. So there are different sequencing. Still, the word chemistry is being used because everyone understands chemistry. Because RNA has lots of modification. See, for DNA already have reached Q20, Q25. So RNA, now the new chemistry in quotes, is increasing the accuracy to almost close to 99% accuracy. So this native RNA modification, M6A or other modifications, everything, there are software available or they call models available. And there is no need for harsh chemical treatments like bisulfite. You can directly sequence them. So here is one example how a bacterial transcriptome and epitranscriptome both have been deciphered using Minion sequencer. Now we are usually using Promethean sequencer, which has almost 5 times, like it can generate almost 150 GB data from each flow cell. So in all these cases, we have to do ribodepletion. Otherwise, 90%, 95% of the reads will come from the ribosomal RNA. That's true with any type of RNA-seq. So where do we apply this transcriptome sequencing in agriculture? See, there are lots of applications in biopharma, vaccines, that I'm giving a list of papers for you to go ahead and read. So beyond SNPs, agriculture is almost everyone does SNPs, and then they go and do PCR-based SNP assays. So beyond SNPs, a lot can be done in agriculture using RNA-seq or transcriptome sequencing. First is you can quickly get SNP markers when a genome is not sequenced, and you have different lines with different traits. You can sequence the RNA alone in wheat, actually, for even for bulk segregant analysis, because a wheat genome is large and it's cumbersome to do whole genome sequencing. It's easier to do a RNA-seq and then find the SNPs. And these SNPs markers, because they are all lying on actually relatively higher copy transcripts, these markers are more valuable than random or arbitrary SNPs which are occurring on the whole genome. And secondly, expression as a marker for traits. If you want to go beyond the competition and you want expression as a trait, there was a company called AOGene in Israel, where we were doing we are finding what are the genes which differentiate between the two hybrids. Not the genes, but the gene expressions. And gene expression is also very important to understand hybrids and varieties. See, we are still looking at only SNPs. They only tell the difference between the hybrids and varieties. But what is the gene expression difference, right? That is much more valuable to know. And of course, in agriculture now, CRISPR is approved in many countries for many applications, many traits. So you can develop new targets for CRISPR by RNA-seq. Now, the all-important table comparing short reads versus long reads in an unbiased manner. If you want to do a short read, the price per RNA per sample is around 15,000 to 17,000 Indian rupees. I think this is around $200. In long read, it's slightly more, maybe $250. And depth in short read usually given is around 5 GB of data. These are 100 parent reads. In Nanopore, 3 GB of data. The only disadvantage with the lower amount of data is that very low copy genes, you may not find them, what you might find in short reads. For that, you have to do more sequencing. But it also benefits to do more sequencing because the low copy genes, instead of some pieces lying here and there, you will get full length. And SNPs, if you're doing SNPs, yes, it's good to do short reads. If you're doing transcriptome sequencing to find SNPs and take the SNPs, it's better to do that. Whereas the SNPs you will find only in high copy transcripts in long reads. This is one because the main reason is you will need at least 10x coverage, 15x coverage to find SNPs accurately in a long read technology. Full length, no. In short reads, obviously, yes, here full length transcript you will see, which is the most valuable thing. Gene fusions, it's mathematics, it's possible, but it's very indirect approach because you don't have a reference seq to align and do. Now you have to do a lot of approaches and then prove it. Here it's a direct approach. If there is a fusion, there is a fusion transcript available. RNA modification, you will never find it. Long reads, yes. Poly-A length, you will never know. Here you will know. Splice events, yes, but it is going to be, I put no because you cannot authentically say certain splice event has not happened. What has happened, maybe they will find some of them. The frequent splice events it will find, but since you don't see it in a single RNA, it's going to be difficult there. Whereas here you can directly find it. So both of them are suitable in two different places. That is, if it's a tag-based single-cell RNA sequencing, transcriptome sequencing, single-cell transcriptomics, tag-based means you're sequencing only small tags, the 350 bases or maybe just 100 bases, because you're only counting. That's good. Whereas it's ideal for full-length RNAs, like you want to see what splice forms are done, it's good to go with long reads, those kind of 3' based, poly-A based, or random primer based sequencing. You will need long read sequencing. Analysis of RNA-seq. See, many of the people who do analysis, they have to give it to a bioinformatician. They will come and give RPKF, this KF, that value, this value, and expression values. And that's where the problem comes, whether the RNA gets down-regulated or that splice form gets down-regulated or up-regulated, we never know. Whereas in long reads, it's directly counting. Hey, this is my number of, this is a number of copies of this RNA, and that's the copy. Accuracy is Q30 in short reads, Q20 in long reads. You can increase accuracy in long reads by using a bigger server, but it's not at all required because you are only looking at gene expression. But if you want to look at SNPs, I would still say, why are you using Q30? Please go for Q40. This Q40 is onso-based sequencing, where it's Q40 to Q50 actually. So if you want accuracy, if you're going to look at SNPs or very small indels, even for very small indels, you better do long reads and short reads together. And very important samples, it's better to do long reads. And if you think accuracy is needed because the genome was not sequenced, or you're looking at low frequency modifications like RNA editing or something happening, then please go for Q40 plus to Q50 plus of onso. Why are you stuck with Q30? I think the days of Q30 is coming to an end. It's going to become long reads on one side because it gives the answers directly. And when accuracy and SNPs and all these things go with onso Q40. That's what I would recommend. So here are some important papers I have a list also to give it to you. So this is a Nature article where different human tissues. So here we are not looking at SNPs. We are not looking at even expression. We are looking at the expression of different variants, transcript variants. For example, if you take enzymes, some liver enzymes and muscle enzymes, same enzymes are there. People call it isoforms, right? This isoform is expressed in liver, that is in adipose. So what is isoform? Isoform is nothing but a splice variant, right? So even for viral transcriptome research, it's important to do that. And someone is doing a biopharma, gene therapy, all these cases, it's important to do a full-length RNA sequencing, right? So these are all important publications, which I'm going to share with you in this publications. And then here is a list of important tools and publications. For example, here is one which is called as the dark side of human transcriptome, because many of the genes are still not known, because all these tiny, teeny pieces people couldn't put together. Sometimes they didn't even know from which part of the genome it's coming. So long read transcript sequencing is the way to go. So these are important articles which will be shared with you. And I'm summarizing my 30 minutes getting over. There's going to be a lot of time for questions, I hope. So the summary is, we are suggesting Q40 plus short read whenever you think you will need higher accuracy. And for routine transcriptomics, please use long read sequencing. It's already known, like whole genome, metagenome, and many applications, its long read is far better than that. And then when you come to single cell transcriptomics, we suggest for tag-based or short tags sequences to be done purely for expression. For example, 10x platform has both. So for that, you use Onso. And when you're looking at 3' based libraries on 10x platform, long reads, because you're looking at transcript variants, right? You're not looking at SNPs. You're not looking at expression. Maybe you're looking at expression also, but do you need more on transcript variant, then you use long reads. And then you have BioScript, which we are also distributing it in India, single cell genomics. This is, I think it's important to do long reads and not short reads for single cell genomics. See, apart from this, if quite a few of you have said you're still using microarrays, custom microarrays or standard microarrays, Agilent, we are still certified. We work closely with them, customizing. And going beyond this is SomaLogics. You might have heard. You can actually now almost proteomics like transcriptomics. That easy, like 11,000 proteins of human or mammalian can be assayed. Expression values can be got with just some 50 microliters of culture fluid or blood or plasma. So this is the summary. And thank you all of you. This is our team. I thank also Dr. Kirti, who's sitting here, for putting up this PPT last minute. Many new things were added. It's really hot, what you are getting. And then how to contact. So we have Indian academics, Swapnil, you can reach, Indian corporate, and of course, global business. Dr. Kirti, you can join, you can reach her. There are different projects we are doing. One very important thing, adaptive sampling, is that you sequence only the regions of your interest. This is like if you have if you want only one gene, like BRCA1 to be sequenced, whole gene to be sequenced, you can use in Nanopore adaptive sequencing. Direct RNA, I already spoke. metagenomics. I will never do short reads. See, we are still doing 16S, V3, V4 because it started with Sanger times, Sanger sequencing. That is a length, like 600, 700 bases accurately you can sequence. That is how it started. Then when Illumina MiSeq came, 300 cross 2, which many of you would have noticed, it's never 300 cross 2. It may be 250 cross 2, 270 cross 250 or something like that. So it all got stuck with that, what we can do. Now you can actually sequence whole of 16S if you want in long reads. Then metabarcoding, cDNA sequencing, you already heard. And then panel-based genome sequencing, which means part of the genome only you want to sequence. Now you better sequence the whole genes instead of only sequencing the exons, because short reads can only do that much. And methylation signature, you can simply sequence 2 or 3 human genomes on one flow cell, Promethean flow cell. It actually counts to be cheaper than short reads. And then you get the complete methylation signature also of the whole genome directly. Of course, CRISPR-Cas derived cells or plants, it's important to do long read sequencing. And if you require on those target regions, short reads for high accuracy sequencing. Structural variants, I don't think you will ever get by short read sequencing. Translocations, balanced, unbalanced, all of them only long reads. And single cell transcriptomics and single cell genomics always go for long reads, unless you want a tag-based sequencing. And that too, there we suggest going for Q40 plus. So basically, I'm trying to tell you that this Q30 is neither too accurate nor long enough, but we are stuck with this old technology. I think it's time to move ahead to higher accuracy or higher read lengths. So thank you all of you, and over to you back, Saroja.
Thank you, Dr. Raja, for very informative webinar. We really appreciate you for taking out time to educate us about advantages of long reads for transcriptome sequencing. Now the session is open for Q&A.